All right. Morning, everyone. Afternoon. I never know what to say at this time of day. Okay. All right. Today, we're going to be lecturing a little bit, but a reminder, there is a set of interactive notes that's due today by 5 p.m. on Brightspace Dropbox. That's going to be the Principles of Disease interactive notes then. We're going to be lecturing today. We're going to be lecturing next week. For next week, there is going to be another Brightspace quiz on Wednesday that'll open up after Wednesday's class, go through Thursday to midnight then. That is going to be for the quiz. There will be another interactive note set due next Friday then as well. That'll probably be your final interactive notes that is due though. Okay, so this week's and next week's. The other thing then is coming up as we end here the semester. Remember, fourth exam, which is going to cover kind of this last unit of information. That is going to be on Friday, May 8th. So on Friday, May 8th, 10:30 a.m. in here, we've got our fourth exam. That is going to be in the same format as throughout the semester with a mixture of some multiple choice or matching questions. There'll be the pathogens of interest that are on there. There'll be a short answer question or two on there then as well. So same format for exam number 4. Monday, May 11th, I'm going to have that as a review period. Officially, the last day of lectures is Monday, May 11th. We're going to be in here 10:30 a.m. You guys don't have to come. I recommend you come. But at 10:30 a.m., I'll have a review session for the cumulative final exam then. So just come with questions that you want me to go through. And remember then, the final exam is cumulative. That means anything we've talked about over the course of the semester could be on that final exam. Obviously, it's going to be probably the more important stuff or the things that I've asked about multiple times throughout the semester then. With that, the final exam is cumulative. It's on Wednesday, May 13th at 10 a.m. in here. Okay. And so that is a cumulative one. Remember, the final exam is going to be 50 multiple choice questions. Each multiple choice question is worth 2 points. So it's a 100-point exam total, but it's 50 questions. 40 of the multiple choice questions are going to be cumulative. That means anything covered over the course of the semester could be in those 40 multiple choice questions. The other 10 multiple choice questions are going to be specifically over the pathogens of interest series that we've been going through all semester. So any of the pathogens of interest could be on the cumulative final exam. So make sure you're going back through those in detail. Questions about that? Okay. Also remember, there's an extra credit opportunity. It's not open yet, but the IDEA course evaluation surveys, I think, go out on May 1st. That's a week from today. And those I give extra credit for based upon the class participation rate. If 70% or more of the class fills out the survey, everybody gets 2 points of extra credit. If 80% or more of the class fills out the survey, everybody gets 5 points of extra credit. And if 90% or more of the class fills out the survey, then everybody gets 10 points of extra credit. Questions about that? All right, last lecture we left off talking about then pathogen of interest, C. diff, Clostridium difficile. This is a gram-positive bacteria that causes colitis, which is inflammation and infection of the colon and large intestine then. This is typically going to be severe diarrhea, intestinal inflammation, abdominal pain, and this could last for weeks to months in the case of a C. diff infection. And long-term diarrheal diseases are really bad. It can get a lot of dehydration, lack of nutrition, and this can even be fatal in cases as a result of that. C. diff is usually not going to impact normal, healthy individuals. C. diff is almost always a nosocomial infection or hospital-acquired infections, with most infections following antibiotic usage. Now, we're going to hit this again. We're going to talk about antibiotics specifically, but antibiotics are chemicals that kill bacteria, but they kill all bacteria. Okay, so we left off talking about this at the end of last lecture, When I say antibiotics kill bacteria, yes, that means they'll kill bad bacteria, disease-causing bacteria, but they also kill the good bacteria in your bodies then as well. And the good bacteria, well, they're good for you. They keep you healthy, and they actually can prevent pathogens from infecting and colonizing your body. So after antibiotic usage, yes, you've killed microbes, but you've killed all your good bacteria as well. You come off of the antibiotics, now you don't have that normal microflora there, and that can then lead to a patient being a compromised host, and that can lead to then C. diff. Normally, the normal microflora in your body prevent C. diff infections. After antibiotic usage, you have very low to no normal microflora in your body. Now there's no competitors for C. diff. C. diff, you get exposed to it, you get an infection. C. diff is endospore forming. So it's also an obligate anaerobe. What's obligate anaerobe mean again? Yeah, it cannot grow in the presence of oxygen. If it's in the presence of oxygen, it's in an endospore form. So the if it's in oxygenated conditions, it's in the endospore form. That is the dormant version of bacteria that can survive through harsh conditions. If someone then gets exposed to an endospore, say they ingest the endospore of a C. diff, well, now the C. diff endospore makes its way in the digestive tract. The endospore germinates, wakes up. Our digestive tract has very low to no oxygen conditions in it. So our digestive tract is actually an anaerobic environment. And now the C. diff germinates, wakes up, starts actively growing in the gut. C. diff is also much more prevalent in developed, what we consider kind of the more Western developed countries. Why would that be the case? Yeah, you have higher antibiotic usage. And so typically then with these C. diff infections, again, they're almost always following antibiotic usage. Somebody comes off of the antibiotics, but they've killed all their normal microflora that usually would prevent a C. diff infection. Now, once they're off the antibiotics, if they get exposed to C. diff, they get a C. diff infection. And this is why it's a common nosocomial infection. For instance, even things like surgery patients are almost always put on prophylactic antibiotics. That just means they are given antibiotics to prevent an infection from the kind of surgery. But then they're on antibiotics, they come off of it, they get a C. diff infection, is what tends to happen. Anybody know what the treatments are for C. diff infections? Huh? It is actually not antibiotics. It might be antibiotics in the short term, but if you give a patient antibiotics, what happens is C. diff will just start making endospores. And that is the dormant version of the cell, and that survives through the process. You come off the antibiotic, C. diff just comes right back. / probiotics. Common infections nowadays for this one is what is called a probiotic treatment, but they do what's called a fecal transplant, which is not as bad as it sounds. But what they do is they take actually a normal, healthy individual that has normal microflora in their gut, but they take a sample of their fecal matter. In that fecal matter is all the good bacteria. Okay, and so the good bacteria are in there. They actually dry it down and put it in a pill that either then has to be, why am I blanking on? I am blanking on the term. Oh man, I should have looked this up beforehand. It's either then given orally, so you have to swallow it, or it's given then rectally. What's the suppository? That was the term I was looking for. I couldn't remember. But what they do is they put it in a pill form, but it is just dried down fecal matter, including the good bacteria in it. This is a fairly common treatment for C. diff, unfortunately. They are starting to move to just more probiotic pills instead, as they get more research onto what would be like an effective probiotic pill that you're going to give to somebody with a C. diff infection. But really, what you're trying to do with C. diff infections is introduce the normal microflora that are then going to outcompete the C. diff and get rid of the C. diff over time. Questions about that? Dysbiosis. This is when gut microbes fall outside of a normal, healthy state. This could be too few or not the right species. Okay. So dysbiosis, this is when your gut microbe population is wrong in some way. Either you don't have enough gut microbes or you've got the wrong species of microbes in the gut. This leads to a condition called dysbiosis. This is implicated in various different diseases, including C. diff, as we've kind of talked about here, but also things like Crohn's disease, irritable bowel, IBS, irritable bowel syndrome, and ulcerative colitis, where you get like ulcers in the colon. This is often as a result of like not having the correct kind of gut microbiome in the body. Most of these diseases are also more common in what we consider the Western or developed nations. Again, this is due to a combination of both diet and antibiotic usage. Okay. In these areas, antibiotics are used more often, but that kills your good microbes in your gut, and that can cause dysbiosis. Also, our diet almost tends to be too clean and without enough kind of variation in it. Most of our diets are very grain-based, whether it's things like wheat or corn or rice or things like that. It's very, very heavy in the grain, very limited types of diets in many cases. And so also our food tends to be almost too clean, so we don't get exposed to a diverse set of nice, normal, good gut microbes sometimes. And so this can cause some of this dysbiosis to be more common there, and some of these GI tract disorders are then much more common. There is also a lot of research that shows there's a linkage between the gut microbiome to also obesity, depression, Parkinson's disease, and some other disorders then as well. For instance, you can take microbes, gut microbes, from an obese mouse. And if you transplant the gut microbes from an obese mouse into a skinny mouse gets fat. And so there's a lot of linkage between what kind of gut microbes you have and things also like diet. Turns out a lot of the gut microbes say they like eating fats, or say you have gut microbes that like eating sugars. Then you eat a donut or something like that, a high fat, high sugar thing. They actually make compounds that then give you a good mood. The bacteria make some compounds that make you happy because you ate food that made them happy. And so they can control your dietary preferences, and then you're going to eat more of that sugary and fatty food that the microbes like. They're doing research on that, but it doesn't always work as well the other direction. Okay. By the way, this is partially because you have almost kind of a second brain. You've got most of your nerves and your neurons are in your brain, but the second greatest concentration of neurons in your body is along your digestive tract. And so that actually does control a fair amount of your behavior. And then the bacteria will make compounds that also control your behavior then as well. Like they'll make Valium and some other types of things that are mood-altering drugs. All right. So everything we talked about up to now has been kind of the first lines of defense, the non-inflammatory innate immunity. I do want to talk also still about innate immunity, but now about inflammation. Okay, so things that are involved in inflammatory response. So if microbes get past the initial host barriers of defenses, there are further innate barriers, such as inflammatory responses that give a second line of defense. Part of the inflammatory response is what is called phagocytic cells, or the phagocytic barrier. Phagocytes, these are cells that destroy foreign things in our body by doing a type of endocytosis called phagocytosis. Now, what exactly is phagocytosis? Phagocytosis then is just going to look like this. What happens is you might have a phagocyte cell. That phagocyte will extend out what are called these pseudopodia. Pseudopodia are just these membrane extensions. So it sends out these membrane. Is that the fire alarm? It usually is way more than that. They didn't make it, huh? Well, we are supposed to go outside. So out you go. So, yay, fire alarm. All right, so pseudopodia, these are membrane extensions that go kind of extend out from the cell. So if this is my phagocyte here over here, it's got these then pseudopodia that extend out. They wrap around then a, say, bacterial pathogen or a viral particle. They then bring that into the cell as something called a phagosome. And this is just a digestive compartment inside the cell. The phagosome fuses with a lysosome. Lysosome is actually going to think of this as analogous to a cellular stomach. It basically has digestive enzymes plus acids inside of it. And so this lysosome fuses with the phagosome. The lysosomal enzymes digest that captured material, and in essence, you kill any of those bacterial pathogens then. And then waste products get released out of the cell. But overall, what you have is these phagocytes. Phagocytes include things like macrophages, neutrophils, basophils. These are the major phagocyte cells of our body. These are also all examples of what are more commonly referred to as white blood cells, then, for instance. There's a bunch of different kinds of white blood cells, though. Macrophages, neutrophils, basophils are different kinds of white blood cells, but all of them have phagocytic activity where they'll engulf these bacterial pathogens, these viral pathogens, and then digest them. Then. And that is a major thing that helps protect our body early on during an infection. Anybody ever had like you get a cut or you get some other things that gets infected, you get some inflammatory things and you got kind of that white pus? Yeah, that's white blood cells, and most of that is phagocytes that's trying to go around and gobble up bacterial or viral pathogens then. So phagocytes are going to protect us. They also make things like hydrogen peroxide, nitrous oxide, some other chemical inhibitors to kill pathogens then as well. So they'll secrete out a number of different chemicals, and they use that to kill pathogens. So our body does actually make hydrogen peroxide. These phagocytes will secrete that out towards some, say, viral pathogens, some bacterial pathogens, to try to kill them using that type of defense mechanism as well, a chemical defense mechanism. Now, phagocytes are part of then the inflammatory response. So inflammation is part of the innate immune system, and it will activate a response. And I do want to kind of talk about, this is a summary slide that kind of shows the steps, 1, 2, 3, 4, in an inflammatory response, but I'm going to go through this a little bit more step by step. Inflammation. This is characterized by swelling of an area. the area that's inflamed is going to look red. It's a redness. It's going to feel hotter to the touch than surrounding tissues when you have an inflamed area. There's going to be pain in that inflamed area, and sometimes there's also loss of function. Anybody ever broken in like their wrist or bones or anything like that? It swells up, right? So I've broken my wrist too many times, but it swells up, and it swells up to the point where you can't really bend the joint anymore. Loss of function is actually kind of important because if you keep using the wrist after you damage it, you might make the damage worse and worse. So the swelling, especially of like joints, keeps you from using that joint and keeps you from making the injury worse than it already is. And so that loss of function is part of the then inflammatory response. Now, what happens with the inflammation? So let's say there is some type of damage. So you have damage to tissues in the body. Let's just say maybe it's a cut, for instance. You've got a cut in the skin. Okay, maybe you've gotten cut then. So you have some type of damage to tissues, but that also means then you got some damaged cells. Those damaged cells start making, within seconds to minutes of tissue damage, what are called cytokines. Now, cytokines, these are protein signals that are used to communicate and activate white blood cells and your immune system. So cytokines are specifically these protein communication molecules, these proteins that help to activate immune responses. Okay, so that is what a cytokine is. We're going to talk a couple times about cytokines over the next several lectures. So these are going to come up again and again, and at least a third time. So cytokines control immune responses, including sometimes inflammation. So one of the things that happens is as these cytokines are made by the damaged spot, and they go over to the closest capillary bed. Capillaries are small little things that carry the small little parts of our circulatory system carrying blood. We have lots and lots and lots and lots of capillary beds throughout our body. But what they do is cytokines lead to dilation of the capillaries. So if this is the normal size of a capillary, as those cytokines are then binding to receptors in the capillary bed, they cause the capillaries to go from their normal size to a larger dilated size. Dilation means they get bigger. This then leads to those capillary beds being leakier. Because you have those cells that make up a capillary and line it, because they're more spread out, now when it's dilated, more fluid leaks out of the capillary bed and to the surrounding tissues. This leads to an increase of fluid at the site of injury that is referred to as edema. Edema is just going to be fluid buildup in an area, okay? And that's because these capillary beds get dilated, more fluid leaks out of them, fluid accumulates in the inflammatory site. That means you have more fluid in the area, more blood in the area. Now, what's the color of blood? Red. So what you get is because you have more fluid in the area, you have swelling. Because you have more blood in the area, it turns more red. That's where the redness comes from. Also, you get a lot of blood that's from your central core part of your body. And it turns out your core is warmer than, say, your extremities, like your hands and feet. So if you measure somebody's like external temperature, it's what we call body temperature. Like say you're measuring forehead temperature, that's their external temperature. And that's going to be 37 Celsius or 98.6 Fahrenheit. Okay, but if you measure their core temperature, it's actually going to be 42 Celsius. It's 5 degrees Celsius warmer in your core, inside portions of your body than that. But that means that if you have blood building up that's from warmer areas of your body, it feels hotter to the touch then. Questions about that? Interestingly enough, also, the average body temperature of humans is no longer 98.6. That's actually data from like 80 years ago. And it turns out the average temperature of humans nowadays is actually 98.2. It's gone down in the last like 100 to 50 to 100 years. So evolution in action. We actually have a lower body temp as a human population on average than we used to. And no, I don't know why, but I know it's changed just a little bit. Cytokines can cause dilation of the capillary beds, but they also are going to be a chemoattractant to white blood cells like phagocytes. So what happens is, yes, those cytokines cause the capillary beds to be then dilated. They also are going to cause the phagocytes to migrate to that site of injury. Phagocytes are constantly, those white blood cells are constantly going through our circulatory system. And they're going throughout our body. But when they sense these cytokine signals, they move out of the capillaries, they move into the surrounding tissues, and they migrate to where there was damaged tissues then. Are we okay? Okay. Is something out in there in the lobby? All right. Those phagocytes move to that site of damage, and then they're going to clear out pathogens by phagocytosis. They're also going to start the process of tissue repair then. Questions about that? Now, inflammation is important. Inflammation is a body response that's going to be involved in clearing pathogens and starting tissue repair when you got these damage reasons. However, inflammation can sometimes be acute. Acute is going to be a direct response to tissue damage, and it's usually short-lasting. Acute inflammation is going to combat early stages of infection and initialize tissue repair, and this is a good thing. Okay, so acute inflammation is good. Short, long-lasting inflammation that basically clears pathogens and starts tissue repair. However, there are certain things that can sometimes lead to chronic inflammation. Chronic inflammation, this is where there's widespread inflammation. So instead of inflammation being in just one spot of your body, it's spread systemically throughout your body, or it can be long-lasting inflammation. Chronic inflammation is not good. This can lead to a lot of host immune responses, and then those long-lasting inflammatory responses can actually cause damage to the body. And some diseases that are associated with chronic inflammation include rheumatoid arthritis. Lupus is caused by chronic inflammation, multiple sclerosis, myasthenia gravis, thyroiditis, type 1 diabetes. These all have a linkage to chronic inflammation. Also, chronic inflammation can lead to cancers, something we'll talk a little bit about as well here. Okay. Anybody ever heard that asbestos exposure can lead to lung cancer? If you breathe in asbestos fibers, they make their way into the lung, and our body has no way to get rid of asbestos. Our body can't break it down. So those asbestos fibers just stay in your lung, but what they do is they activate inflammatory responses, and then you have chronic inflammation in the lung against those asbestos fibers for the rest of your life, basically. But chronic inflammation leads to damage to the cells, which can cause mutations, which can lead to cancer and lung cancers then. So chronic inflammation can cause damage to cells, which leads to mutations, which leads to cancer. We'll talk a little bit about that here in a few lectures. By the way, these are all examples also of autoimmune disorders. We're going to talk about those here soon in class as well. Questions about inflammatory responses? All right, another defense mechanism that our bodies have is something called the complement system. And the complement system, this is a group of soluble proteins that circulate around in our bloodstream all the time. And these complement system proteins make up about 5% of all the proteins in your serum. And by the way, serum is just a fancy way of saying blood. Complement proteins are made by liver cells. The liver cells make complement proteins. They secrete them into our circulatory system. Complement proteins are typically always circulating around in an inactive form. But if they bind to specific microbial targets, or if they bind to activated antibodies, this will activate those complement proteins through what is called, don't worry about this too much, through a type of proteolytic cleavage, part of the complement protein gets cut off. That reveals an active site that was previously hidden, and that activates the complement proteins. Complement proteins are kind of both part of innate immunity and also adaptive immunity. So I said there's two main branches of the immune system, innate immunity and adaptive immunity. Complement proteins are sometimes part of innate immunity, but sometimes part of adaptive immunity. So for instance, if antibodies are involved in complement activation, then that's more part of the adaptive immune system. The antibodies are part of the adaptive immune system. Now, complement proteins, when activated, serve a number of important roles in our body. So for instance, if you have a target cell, so let's say this is a pathogen. If complement proteins are activated, they can bind to a pathogen and then they can directly kill pathogens by creating holes in their membranes and lysis of the pathogens. It causes the pathogens to break open. So sometimes these complement proteins can directly damage and directly kill pathogens. So that is going to be one of their functions. Lysis and destruction of pathogens. Some activated complement proteins can do that. They can do lysis. They can also do something called opsonization. Opsonization, complement proteins can act as opsonins. This means that they promote the phagocytosis of whatever they are attached to by a phagocyte. So if you have a bacterial pathogen, so here we have a bacteria. The bacteria is the cell in green here. I have it bound by complement proteins in red. This is then going to be my phagocyte in yellow. This phagocyte is 1,000 times better at phagocytosis if activated complement proteins are on the pathogen. Okay, that phagocyte can actually do phagocytosis of, say, bacterial pathogens or viral pathogens, even if complement proteins aren't attached to it. But it's way better at it, 1,000 times better at it, if complement proteins are bound onto the pathogen. Questions about that? Phagocytes actually have receptor molecules on the surface that recognize activated complement, and then they'll target anything that has activated complement proteins attached to it. Third function, so again, functions of complement system include lysis, opsonization, but also activation of inflammatory responses. So the third function of the complement system is that once activated, complement proteins can activate inflammation. That's going to activate inflammatory responses and then phagocyte recruitment. Questions about that? Complement is a major barrier of our immune system that especially helps to fight off diseases early on in an infection. Now, I also said that complement protein is kind of sometimes part of innate immunity versus sometimes part of adaptive immunity. Innate immunity is what we've been talking about up to this point in this unit. Innate immunity, pretty much every type of multicellular organism has a version of innate immunity. So animals have innate immunity. Plants have innate immunity. And in fact, there is a lot of plant innate immunity responses that are very similar to animal innate immune responses. So pretty much all multicellular organisms have a version of innate immunity. Innate immunity is also going to be providing an immediate response. This is going to be, it's going to be either preventative or it's going to be within seconds to minutes of exposure to a pathogen. So this is going to be an immediate response. It's either about keeping the pathogen out of our body or immediately clearing it. However, it's a broad general response. It just is about prevention or clearance. Like phagocytes, they just target anything that's not supposed to be there. There's not a very specific component to it. Well, and this will make more sense as we talk about adaptive immunity. The other thing, though, with innate immunity is there is no what is called memory response. What I mean by that is innate immunity, the first time it sees a pathogen, it works. The second time you see a pathogen, it works the same. The third time you see that same pathogen, it works the same. Okay? It works exactly the same every time you get exposed to a particular disease. Now, what we're going to start talking about is instead adaptive immunity. Adaptive immunity is really only in vertebrate animals and mostly even then just in mammals. Adaptive immunity is mostly a mammalian-specific immune system response. It's slower. Full adaptive immune response is going to take up to 2 weeks. Okay, so it's not going to be an immediate response. Usually takes at least a few days to activate, and a full, strong adaptive immune response can take up to 2 weeks then. It is highly pathogen specific. So what I mean by that is it's targeting not just any general pathogen, it's targeting very specific molecules that are called antigens on a pathogen surface. It is very, very, very specifically, it's not even targeting a cell, it's targeting one very specific protein or polysaccharide sugar on a pathogen cell. Not the entire cell itself, one little piece of it is what it's targeting. So it's very specific on what it targets, but it also generates a memory response. And we're going to talk about what a memory response here is. But the first time your body sees a particular disease for the first time, adaptive immune system doesn't work very well. If some time passes, you get better. Some time passes. The second time you get exposed to the disease, though, your adaptive immune system responds much faster, much stronger, and it often fights off the disease before you even know you got exposed to it. So a memory response is where a second exposure to the pathogen gets a very strong, very fast response that clears that pathogen before it causes issues. Okay. Adaptive immunity. The first time you're ever exposed to the disease, it takes about 2 weeks to mount a full adaptive immune response. Sometime you get better. Some time passes. Secondary exposure elicits or causes a much fuller, much stronger response, and it takes, instead of 2 weeks, usually a couple days to a week. So it's faster as well. And I'm just showing this here is basically if you have some type of initial exposure to an antigen, it takes maybe 14 weeks to have like a full antibody response. IG stands for immunoglobulins, which stands for actually just another name for antibodies. This is then going to be antibody concentration on my y-axis. But here you can see after an initial exposure to the antigen, very first time you've seen it, you get about this many antibodies made. And it takes about 2 weeks to have all those antibodies made at a strong level. If you have some time pass, so this is a break in my axis here, but it means some time has passed, months, and you get a secondary exposure, well, then you get a much stronger antibody response. You get way more antibodies produced on that secondary exposure to that antigen, and they're produced quicker in response to that then as well. And this is referred to as a memory response. All right, antibodies are part of our adaptive immune system. Antibodies are proteins that are made by the immune system, and they bind onto what are called antigens. Okay, antigens are then specific molecular patterns that are found on pathogen surfaces. I put bacteria here, but I'm going to go a little bit more general. Just pathogens in general. Okay, could be a viral pathogen, could be a bacterial pathogen, could be a malaria, which is caused by a eukaryotic pathogen, for instance. but the antigens are specific patterns found on a pathogen surface. Now, antigens, this has come up already once before, coming up again. Again, antigens are structural patterns on the surface of a pathogen. And almost always, it's some type of protein shape or sugar shape. So antigens are almost always some type of protein that is on a pathogen surface or a polysaccharide sugar that is on a pathogen surface. And any protein or sugar that is foreign to the body, not a normal thing that's in our body, that can potentially act as an antigen that is then targeted by antibodies. Don't get antibodies confused with antigen. Antibodies are the proteins that our immune system make. Antigens are proteins or sugar shapes on the surface of a pathogen that our immune system targets. Questions about that? Okay. We have cells in our body called B cells or B lymphocyte cells. A lot of times they're just referred to as B cells for short. The function of B cells is antibody production. So B cells' job in our body, these are a type of white blood cell, and their job is just make antibodies and make a lot of antibodies. Okay. That's the only thing they do. They're specialized for antibody production. So these B cells will make antibodies. They'll secrete out these antibodies. Antibodies will bind onto antigens on a pathogen surface. This activates the antibodies, which then activates an immune response. Antibodies. When the antibodies get activated, when antibodies bind onto antigens, that activates them. Activated antibodies can then activate the complement system, and then the complement system destroys the pathogen. Activated antibodies can act as a opsonin. Again, that means they're going to promote phagocytosis of whatever they're attached to. And they can also activate something called antibody-dependent cell-mediated cytotoxicity, or ADCC for short. I'm not going to talk about that too much. We're mostly going to talk about this kind of role for antibodies as we go through some things, the first two. But this is where a T cytotoxic cell or natural killer cell comes in and then destroys whatever the antibody is attached to. But they do it differently than, say, complement system or a phagocyte, so it gets a different name. Don't worry about it too much. We're not going to go into that level of detail. However, another main role for antibodies is then activation of B lymphocyte cells. B lymphocyte cells make antibodies, but also antibodies can be an activator for B lymphocyte cells. And they're involved in generating a memory response. All right, so how does that work? And I'm going to take a closer look at point 4 here. B lymphocyte cells are one part, they're not the only part, but they're one part of our adaptive immune system. B lymphocyte cells will also exhibit immunological memory. Immunological memory, this means that a second encounter with an antigen will cause a quick, fast response, a quick, strong, specific response. Okay. B cells are, again, they specialize. I already went through this a little bit, but they specialize in antibody production. But it turns out we have millions of different B cells in our body. Millions of different B cells. They're all very similar, but they're all slightly different. Each B cell makes its own unique version of an antibody. Have you guys heard that no two snowflakes are alike? Well, then that is going to apply to also antibodies. No two antibodies that are made by different B cells are alike. We have millions of different antibodies that recognize millions of different antigens in our body. Each B cell makes its own unique version of an antibody that binds to its own unique antigen. Now, what's that mean? What I mean by that is we have the B cells. B cells are called B cells because they mature in our bone marrow. B cells for bone marrow maturation. Each B cell creates its own unique antibody. So B cell number 1 makes antibody number 1 that recognizes antigen number 1. B cell number 2 makes antibody number 2 that recognizes a different antigen number 2. B cell 3 makes a different antibody that recognizes a different antigen, antigen number 3, B cell 4, so on and so forth. These B cells are made in the bone marrow, and then once they're mature, they're released out of the bone marrow into our blood, and they circulate around in our body. At this point, they've never seen their antigen. Okay? They're all made at random. They circulate around our body. If a B cell never encounters its antigen, nothing happens. And actually it dies after 2 weeks. So if a B cell never sees its antigen, circulates around our body for 2 weeks or so, and then it dies. If it does encounter its antigen, it goes through a process called clonal selection and clonal differentiation. What ends up happening is it looks like this. Let's say B cell number 1 never sees its antibody, dies. B cell 3 never sees its antibody, dies. B cell 4 never sees its antibody, dies. Let's say B cell 2 does end up encountering its antigen. So there's a pathogen carrying antigen 2, then sends a signal into the B cell that then activates it. It causes rapid proliferation. It goes through rapid cell division and makes many, many copies of itself. And then that activated B cell copies are going to differentiate into what is either called memory cells or plasma cells. And this is where we're going to end for today. I'll pick up with this on Monday. But as that proliferation occurs, some B cells form into plasma cells, some into memory cells, and it's these guys that do memory responses later on. Okay, but we're going to pick up with that on Monday. Oh, exams. I do have, sorry, I forgot about that. I do have exams to hand back today.